Hey guys, I wanted to provide you with this resource so you have a little bit more structure when you're figuring out those cereal dilutions. I'm going to be posting some other resources for you to look at too, but remember it's very important for you to kind of work your way through them, go through that productive struggle, and kind of start to figure these things out in your head before we get to lecture on Tuesday and lab next Thursday. So when we were in class yesterday, we were talking quite a bit about this idea of raw milk and how it might affect pregnant people, as well as kind of using fecal bacteria as a proxy for exposure to disease-causing organisms. So when we're thinking about pasteurization, we're thinking about this process of getting rid of coliforms. What we mean by coliforms are, specifically in this case, fecal coliforms. These are types of bacteria that are often found in feces. They're a big component of the mammalian gut. We have them. The issue is when they end up where they're not supposed to be, or if they're not in the right concentrations. So if they're throwing those communities off balance, they can cause very severe infections. If we see them in our food, for example, if we see them in our milk, that usually indicates that the milk was exposed to feces at some point, and it's probably not safe for human consumption. When we're talking about coliforms, it can mean a lot of different things again. Remember that there's lots of different shapes of bacteria, those individual cell morphologies. Oftentimes we think about cocci, spirillum, and bacillus. When we're looking at those rod-shaped organisms, a lot of the ones that you encounter in lab tend to be gram-negative. That being said, there are a lot of bacilli that are gram-positive, so it's not an equal relationship. But when we're thinking about coliforms, these are gram-negative rod-shaped bacteria. When we're thinking about fecal coliforms, these enteric bacteria that live in your gut, these tend to also have the ability to ferment lactose. So they are able to take that lactose sugar, go through fermentation processes, and survive and generate energy that way. So we want to kind of measure the amount of fecal coliforms to determine whether food is safe or not. Remember that with pasteurization, it kind of messes up the cell shape, but it does kill the bacteria. It doesn't remove them necessarily from the culture or the milk. So we can look at it under a microscope, but have no idea how many of those are actually alive and able to cause disease. The reason we want to do what we call viable plate count, putting that culture onto a plate and trying to grow it up, is because that tells us, one, are these the type of bacteria that are likely to cause disease? And two, are they actually alive? Do they have the potential to cause disease if they make it into our bodies? We also talked a little bit about this idea of finding a sweet spot when we're plating cultures. We talked about this in the context of bacterial growth. And I asked you guys which of these you would like to plate and count. So if we're trying to get good data and also make sure we do a good job, we want to try to count the plate that's in the middle, that sweet spot. So the way that we get that sweet spot is we do what's called a serial dilution. Serial means like a series or kind of an ongoing process that's very consistent. Dilution is diluting the sample. So we're going to be taking a very specific amount of the original culture. Usually an easy example is taking 1 milliliter of the original culture and adding it to 9 mils of a blank. Usually with this, we want to use the culture medium as our blank, not necessarily water. Sometimes it's easier to visualize with, for example, food coloring in water. This image and this link takes you to kind of an interactive website if you want to explore this more. When we're thinking about particles here, what we're really thinking about is the number of cells, specifically viable cells that are able to grow on a plate or cause disease. So every time we do this, we're going to be diluting the sample, for example, 10-fold. There's the original culture. We take 1 ml of that, add it to 9 ml of culture medium to make the next in the series, and that is 10 times as diluted. But then we're going to take 1 ml of that dilution, add it to 9 ml of blank of the culture medium, and then it's even more dilute. So the first tube after the original culture is a 10-fold dilution, but then the next tube is actually a 100-fold dilution relative to the original tube. So we're able to kind of take these small amounts and get like 1,000-fold dilution, even though we're only taking a very small amount of the original culture. So this is really useful because we can grow up a lot of very dilute culture. It's actually easier to kind of have a small amount of concentrated culture. Remember, bacteria need to do stuff like quorum sensing and figuring out who's around them. They have to go through that lag phase before they get into exponential growth, but exponential growth is where you want to study them. And it's very challenging to get them into that exponential growth phase if they're in a very big vat, like a bioreactor. It's easier to just grow up a small amount in a flask and then dilute it to get the volume of culture at the right concentration that you need. It also makes sure that when you plate it, you're able to, one, know how much you diluted it, and two, find the right kind of distribution of viable cells that is kind of, we know exactly how it relates back to the original culture. So we're able to do that accurate count and also do the math, do some calculations to figure out how that reflects back on the original culture. So in this example that we were looking at in class, when we raise anything to a 0 exponent, it just means 1. So for example, this original culture, 10 to the 0, that's not diluted at all. It's just 1. But the next one, 10 to the minus 1, that's a 1/10 dilution, or a 10-fold dilution. The next in the series is 10 to the minus 2. That's a 100-fold dilution, or 1/100. And again, we're doing this so that we can find a plate that has that good 30 to 300 range of colonies, that's precise and accurate, and that we can then figure out something about the original culture. So when we're trying to solve different problems, one of the things you might need to do is figure out that exponent. Figure out, you know the volume of your first sample, you know how much you've diluted it. What is your dilution factor? So this kind of ties together that number, that mathematical kind of component, that exponent, with what you're doing in the lab. And I think that's really important. There's a lot of math here, but it really just reflects what you're doing in the lab. What are you as a scientist, as a microbiologist, actually doing? So for example, if you're studying raw milk and you dilute 1 milliliter of milk into 99 mils of sterile water, so you're taking just 1 milliliter of that original sample and putting it into 99 mils of sterile water, what is our dilution factor? To calculate that, we take the amount of the first sample, 1, and divide it by the total volume, so 1 mils of milk and 99 mils of sterile water. That means we are going to divide 1 by 100, and in scientific notation, in an exponent format, this is going to be 10 to the minus 2. So if we need to figure out our dilution factor, if I'm asking you to find the dilution factor from just how much raw inoculum we had, this is the type of calculation we would use. The numbers might change, but this is the general equation and format. So we can say that this is a 10 to the minus 2 dilution factor, or a 100-fold dilution. The reason I want to specify that is because if you look up these formulas online, you're going to see some kind of variations in how they're formatted. I really want you to get in the habit of writing down dilution factors, not other formats, not 100-fold dilution. Just get used to saying 10 to the minus 2. So sometimes we can kind of skip ahead if we only have certain pipettes, or we need to do this very quickly and we can't do a very long series. We can skip ahead in the series. And in order to calculate that new dilution, what we need to do is look at the product of those other dilutions. So for example, if I have 1 milliliter of my 10 to the minus 2 dilution from that previous problem, and I have a 99 mil blank, I have 99 mils of sterile water. If I add that 1 mil to that 99 mils, it's not going to be the next number in the series. I'm skipping ahead. So I need to take my dilution of the sample that I'm doing right now, so that part of the previous problem. So taking the total volume, it's going to be 1 mil out of 100 mils. That's 1 mil of this new sample divided by 1 mil plus 99 mils of the sterile water. And I'm multiplying that, I'm finding the product of that, and my dilution, the sample that I'm actually working with, that's going to be the dilution factor of my new sample. So here, this is the dilution of this new thing that you're doing by kind of diluting 1 ml into 99 mL. This over here is referring to the problem on slide 7. So how you got this actual 10 to the minus 2 dilution. So when you do that, when you multiply exponents, you actually add them. So if they have the same base, you don't have to change anything about that 10. What you can do is take minus 2 and minus 2, you add them together, and you get minus 4. The reason I wanted to specify that is because minus 2 times minus 2 is positive 4, so you know here that you're adding them, you're not multiplying them, but it is tricky because you're multiplying the base. So when you multiply those bases and they have exponents, you're actually adding the exponents together, you get minus 4. So that's tricky, but that's how you solve that dilution problem. So this is useful for figuring out how much you've diluted something, but remember we want to work with actual plates. So when we're doing that, we're trying to oftentimes figure out the colony forming units, that potential to form colonies, the cell density of living cells in the original sample. And we do that by taking what we can observe in the lab, the number of colonies that grow up on that plate, how many we count, and dividing that by how that plate was prepared. So if we know that we added more volume, then it's not going to be as significant, right? But if you added a very small volume and you got the same amount of colonies, that says something. So you're kind of controlling for adding that volume. And that volume has also oftentimes been diluted. So the dilution factor, again, is how much you diluted that serial dilution relative to the original culture. If you haven't diluted it very much, then again, it's not going to be as significant, this number of colonies. If you've diluted it way far and you still get like 300 colonies on the plate, that's a really big deal. That means you have a lot of colony forming units. So again, to visualize this, so it's going to be the same exact formula, but just with visuals that you'll see in a lab. If you take the number of viable cells in that sample, the CFUs, colony forming units, or cell density, that's maybe the number of viable cells in your culture or in your raw milk. You're going to calculate that by counting colonies on a plate. This example is with a type of medium called MAC, or McConkey agar. You're going to work with that a lot this semester. And you're going to divide that again by the volume of culture that you put onto that plate times the dilution factor. So I hope when you're going through these problems, you can at least set these up using this resource. So just kind of work your way through them, read the problem, see what it's telling you, and use that information. You can talk to each other, you can write on the discussion board, you could ask me for help, but I want you to at least try these problems before we get to class on Tuesday.